everybody. My name is Noah Enoch, and the discipline that I focused on this semester was the CRISPR discourse community. So CRISPR, or CRISPR-Cas9, as it's normally called, is a gene editing tool that's based on the natural immune system of bacteria that's highly valued for its precision and accuracy in comparison to other gene editing tools. CRISPR consists of two main steps: a guide RNA, known as a gRNA, which marks a DNA strand to be cut, and a Cas nuclease, which recognizes that gRNA and cuts the DNA. After cutting the DNA, the scientists can influence the way that the DNA repairs itself in a process known as homology-directed repair, or HDR. This is what allows the scientists to actually code for traits. So what makes CRISPR so useful? The ability for scientists to code for desirable or undesirable traits is what makes CRISPR so useful. They've already been able to use this to do things such as increase crop growth, yield, nutritional value, and hardiness, as seen on the image of the right. When they decreased a certain gene found in corn, they actually were able to increase the kernel size. Alongside the editing of crops, CRISPR is also already being used in the treatment of chronic viral illnesses or genetic diseases. The CRISPR community consists of a mix of scientists, medical professionals, ethicists, and government bodies, all united by the shared goal of making gene editing more efficient, affordable, and safe. Scientists and medical professionals work with CRISPR technology to conduct research and run trials, while ethicists and government bodies such as the FDA are tasked with creating laws and policies for these researchers to follow to discourage unethical or exploitive experiments. Communication within the CRISPR community is comparable to most scientific communities and generally consists of scientific journals or collaboration through research trials. Communication between members of the community and outsiders is different and is mostly concerned on the ethics and regulation of CRISPR technology. Alongside unique methods of communication, members of the CRISPR community have developed a unique lexicon that serves a purpose, preventing those unfamiliar with the community from interacting it, while simultaneously making it easier for members within the community to interact with each other. By having unique phrases to communicate with, the CRISPR community has set itself apart from other scientific communities. Issues present in the CRISPR community can be split into two main sections: scientific and ethical issues. Scientific issues that the CRISPR community faces includes the risk of off-target effects, the breakdown of CRISPR-modified cells, or the inheritance of CRISPR DNA, which is known as germline editing. Ethical issues that the CRISPR community faces includes unregulated experiments, accessibility and cost, lack of long-term safety research, and the pursuit of desirable traits. Incident is a phrase used to refer to the actions of He Jiankui, a scientist in China who revealed the creation of the first CRISPR-modified babies. This is what sparked the main global conversation on the ethics and safety of CRISPR research, with many countries implementing new policies to avoid a similar crisis. My interest on CRISPR technology initially came from the controversy that surrounded this technology. Researching the community has only increased my interest in various parts of the community. I'm now interested in the relationship between the CRISPR community and its governing bodies, CRISPR's ability to cure chronic genetic and viral conditions, and the quantity of untapped potential that the technology has. Given that it's such a new technology, the future of the CRISPR community is bright, with many of the things that it can do barely being discovered. In order to implement CRISPR and make it more accessible to the public, members of the community need to focus on addressing the risk of off-target effects, the manufacturing of drugs that utilize the CRISPR-Cas system, the creation of a gene monitoring system to track CRISPR-modified cells, and make efforts to improve and maintain relationships with the public by addressing ethical concerns. Here's a works cited.